the last speaker of this session. It's Vanessa Castelli from Italy. And the talk is about PARP selective antagonist GW6471 that inhibits cell-growing breast cancer stem cells. And I would like to ask Vanessa if she can start to share the screen. Perfect. If you just click on the presentation. Yes.
Okay.
The floor is yours.
On the first one, okay. Thank you for your kind introduction, the chance to present part of my preliminary data here. Breast cancer is the most diagnosed cancer in women and the second leading cause of cancer death. And 50% of triple negative breast cancer accounts for approximately 50% of breast cancer. And this is a kind of cancer that differs from other breast cancer because it lacks of the receptors commonly found in breast cancer, including estrogen and progesterone receptor, as well as HER2 protein. Indeed, this group of patients did not respond to hormone therapy and neither chemotherapy, showing a high risk of recurrence and death. Cancer stem cells represent a small population of tumor cells, and it has been demonstrated that they show a crucial role in tumor formation, progression, metastasis, and recurrence, as well as drug resistance. Thus, they can represent a potential target for breast cancer therapies, in particular triple negative. To this purpose, we isolate breast cancer stem cell from a human triple negative breast cancer cell line, MDA MB231, and the isolated mammospheres were characterized mammosphere by clonal selection were characterized by stemness marker and morphological features. CD44, ATP binding cassette subtype G2, and aldehyde dehydrogenase 1 are commonly used to characterize the breast cancer stem cell from a subpopulation. And interestingly, our mammosphere show high level of CD44 and ALDH1. In particular, for ALDH1, we analyzed also the enzymatic activity by aldefluor assay, and we observed an increase in the enzymatic activity of mammosphere compared to evaluate the PPARs in our mammosphere, which are steroid hormones receptor that trigger the expression of numerous target genes, including those involved in glucose and lipid metabolism, but they show also a crucial role in cancer. They can be activated by endogenous ligands such as fatty acid, but also by pharmacological agents. In line with previous published papers, our mammosphere show the expression of PPARs at various levels. However, the isotype alpha is most abundantly expressed at nuclear level, suggesting its activation. For this reason, we treated our mammosphere with a GW6471, and we focused on cancer metabolism and proliferation. First of all, we assess dose response curve with different time points and concentration, and we noticed that GW led to a decreased viability in a dose-dependent manner. In particular, the concentration of 8 micromolar for 72 hours was chosen for the following experiment. Interestingly, from the contrast phase images, we can appreciate that the GW-treated cells show a spheroid size reduced compared to control mammospheres. Cell cycle analysis by flow cytometry indicated that GW-treated cells show a stage of cells blocked in G1 phase. These results is also confirmed by Western blotting analysis in which cyclins D1 and B2 are strongly reduced by GW treatment parallel with an increase of P21 protein level, thus suggesting a cell cycle arrest. These data were also confirmed by fluorescence of both cyclins, and we can see from the figure that the expression of both cyclins is strongly used upon GW treatment. We can appreciate also a translocation from nuclei to cytoplasm, thus suggesting a reduced activity of both cyclins. Cyclin D1 can be regulated by beta-catenin. For this reason, we evaluated this pathway, which is crucial for triple-negative breast cancer. And interestingly, we noticed that GW led to increased level of the active form of GSK3-beta, which in turn phosphorylate beta-catenin, inducing its degradation. This result was also confirmed by beta-catenin immunofluorescence, and interestingly, we observed that central mammosphere beta-catenin is accumulated at cytotic level, while GW-treated cells show this marker at membrane level. And this data, these results is really encouraging because nuclear and cytosolic accumulation of beta-catenin are usually associated with reduced overall survival in all breast cancer patients. To evaluate the effect of the antagonist on mammosphere metabolism, we evaluated the expression of AMP kinase, which is a cellular energy sensor that mediates metabolic homeostasis under environmental stress conditions. Interestingly, we noticed that GW was able to increase both the total and phosphorylated form of this kinase. This kinase, as you can see from the figure, is involved in different pathways, not only lipid and glucose metabolism, but also in the regulation of autophagy. AMP kinase can inhibit cholesterol biosynthesis, thus we evaluate the cholesterol content, a main component of lipid droplets, by thin layer chromatography. As you can see, the level of the cholesterol content are significantly reduced upon GW treatment. In agreement, the rate-limiting enzyme of mevalonate pathway, analyzed by real-time PCR, were resulted strongly reduced upon GW treatment, suggesting a block in the mevalonate pathway. GW6471 was able also to reduce the lipid droplets content, as you can see from the body pie marker analyzed both by immunofluorescence and flow cytometry. And lipid droplets are commonly associated with malignant phenotype, and in particular in breast cancer, a high content of lipid droplets at cytoplasmatic level are related with higher malignancy. Regarding glucose metabolism, the glucose transporter GLUT1 and the hexokinase 2 indicate an inhibition of glycolysis upon GW treatment, usually upregulated in tumors. These results indicated that PPAR-alpha inhibition led to an increased lipid droplets consumption, parallel with decreased cholesterol synthesis and glucose uptake. In line with AMP kinase results, we observe an induction of autophagy level of LC3 protein and parallel with the decrease in P62 protein level. These results were further confirmed by LC3 immunofluorescence, and interestingly, we noticed that GW-treated cells show a high presence of punctate fluorescence, peculiar of the mechanism of autophagy, indicating its activation. Autophagy it has been demonstrated to play a crucial role in cancer biology and therapy, in breast cancer biology and therapy. However, you know that it plays a dual role in cancer. Indeed, it may positively, depending on the context, it may positively or negatively influence tumor growth and cancer metastasis. Regarding cancer stem cell, there is an imbalance in the pathway autophagy-mitophagy, that modulation of this pathway can influence cancer stem cell invasion, migration, immune and drug resistance. In contrast with PPAR alpha, the isotype gamma is indicated as anti-proliferative. Thus, we evaluated the effect of the antagonist on this receptor, in the expression of this. We noticed that GW led to increased level of this receptor on nuclear level, as you can see from the Western blotting analysis, but also from the immunofluorescence, indicating its activation. Furthermore, its gene target CD36, a fatty acid transporter, resulted significantly increase upon GW treatment. Summing up the results collected so far, GW6 potent PPAR alpha antagonist trigger a metabolic stress and energy imbalance, inducing reduces glucose uptake, parallel with an increased lipid droplets consumption and fatty acid transporter. Basing on these results on metabolism, we evaluated the functionality and localization of mitochondria, since they play a crucial role in tumor cell reprogramming. Interestingly, GW did not alter mitochondrial functionality, as you can see from mitochondrial membrane potential analyzed by JC1. However, the morphology and localization are altered by GW treatment. Indeed, from immunofluorescence figure, you can see the dual marker actin mitotracker and the yellow, the colocalization. And we observed that mitochondria in control mammosphere are localized as cell periphery close to lamellopodia, while in GW treated the mitochondria are absent at cell periphery. Based on these preliminary results, we can say that fused mitochondria are necessary for the energetic support for migration and invasion. Our mammospheres, control mammospheres, show a higher migratory capacity compared to GW-treated cells. To better dissect the migratory capacity, we evaluated proteins involved in cell migration by Western blotting, including RHO-A, RAC1, and CDC42, and we observed a reduction of this protein, thus suggesting a reduction in the migratory capacity. In conclusion, GW6471, a specific and potent PPAR alpha antagonist, trigger a metabolic stress and energy impairment parallel with an inhibition in cell proliferation and migration as spheroids formation. Taken together, these observations point towards the consideration that PPAR, to its pleiotropic effect on metabolism, may be an important therapeutic target for breast cancer and other kind of cancer that use fatty acid oxidation as metabolic strategy. On this basis, we propose GW6471 as adjuvant agent to gold standard therapies for breast cancer, opening a possibility to test class of compound in preliminary and preclinical and clinical studies, and future studies will be devoted to test a gold standard therapy for breast cancer, paclitaxel, in combination with GW, and we will perform also in vivo studies. I would like to thank my team and you for the attention. Thank you.
Thank you very much, Vanessa. There were some the sound was not very good from time to time, but I think it's okay that we understand you very much. Yeah, maybe the connection was not so good. So if anybody has any question for Vanessa, please raise your hand or type it into the chat. And I have one question. You said at the end that you're planning to do the further studies on the preclinical level. And this was actually my question. Are you planning to go further? What are the concentration? Is it maybe this very promising inhibitor, this GW, used for treatment of any other cancer? What are the progress? Because the results look very promising.
Yeah, the results are very encouraging, and previously other research group reported the effect on kidney cancer cell. That's why we would like to better dissect the effect in breast cancer. And we would like to test the combination with the gold standard like paclitaxel, and to see if can in some sense help in reducing the invasion and the recurrence of this kind of cancer.
Okay, thank you very much.
Thank you for the question.
Yeah.